Hello and welcome to the stem cells revision video from scienceworksheets.co.uk. Please like, subscribe and comment to support the channel. A stem cell is an undifferentiated cell which can produce more cells of the same type and from which certain specialized cells can form due to differentiation. There are two main types of stem cells in animals: embryonic stem cells and adult stem cells. Embryonic stem cells are found in the early stages of the embryo. An embryo is a ball of cells that is formed once a zygote, which is a fertilized egg, divides by mitosis. So in the diagram, when the sperm cell and the unfertilized egg cell fuse during fertilization, a zygote is formed. The zygote will then undergo mitosis to form a 2-cell embryo, which can divide by mitosis to form a 4-cell embryo, and so on. Embryonic stem cells are known to have the potential to form most cell types by differentiation. Adult stem cells are already partially differentiated, so they can only form limited cell types. Adult stem cells in the bone marrow can differentiate to form specialized cells such as blood cells. Stem cells in plants are found in the meristem tissue. Meristem tissue in plants is found in regions where growth is actively happening, such as the tips of roots and shoots. Meristem tissue in plants can differentiate into any type of plant cell throughout the life of the plant. This is different to animals. Once a stem cell in an animal has differentiated and formed a specialized cell, it will stay as that cell type. Plants can be cloned through cuttings because the meristem cells can differentiate to form specialized cells throughout the life of the plant. This allows meristems to produce clones of plants quickly and economically. Rare species of plants can be cloned to protect them from extinction. Banana trees, for example, are already cloned. Crop plants with special features, such as disease resistance, can be cloned to produce large numbers of identical plants for farmers. This way, all of the plants will have the advantageous characteristic. Stem cells can be used in medicine to treat conditions such as diabetes and paralysis. Embryonic stem cells are the ideal cell type to try and treat these conditions. Currently, stem cell treatments are being explored experimentally for conditions like diabetes and paralysis, though they're not yet standard practice in mainstream medicine. The use of stem cells has potential risks, such as the transfer of viral infections, and some people have ethical or religious objections to their use. At the moment, all of this type of work is experimental, but in the future, we could see treatments like this. Diabetes and stem cells. Diabetes is a condition where the blood sugar level cannot be controlled because the pancreatic cells do not produce enough insulin. Embryonic stem cells would undergo differentiation to form a type of pancreatic cell which produces insulin. And these insulin-producing cells would then be transplanted into the patient, and the patient would then produce normal levels of insulin and would no longer suffer from the diabetes. Paralysis and stem cells. Paralysis can occur when the nerve cells in the spinal cord are damaged, such as during a car accident. Embryonic stem cells could undergo differentiation to form nerve cells, which could then be transplanted into the spinal cord to replace the damaged nerve tissue. In therapeutic cloning, an embryo is produced with the same genes as the patient who suffers from a disease. So in this example, the patient suffers from diabetes and cannot produce insulin, and we're going to use therapeutic cloning to show how the patient can be treated. The woman on the left will supply an unfertilized egg cell. The nucleus is discarded, leaving an enucleated egg cell. The man on the right, who needs treatment, will supply a body cell. The nucleus is extracted and the body cell is discarded. The extracted nucleus is inserted into the enucleated egg cell. This egg cell now behaves like a fertilized egg cell, so it divides by mitosis to form an embryo. The cells of this embryo are genetically identical to the body cells from the man, because the man provided the nucleus. This embryo is grown in a petri dish. Hormones are used to make the embryonic stem cells differentiate into specialized cell types, such as pancreatic, insulin-producing cells, and these cells are then transplanted into the man's pancreas, where they produce insulin. So in theory, he will no longer have diabetes. As the transplanted cells are genetically identical, they will not be rejected by the body's immune system. Benefits for the use of stem cells in medical research and treatments. Treatment of diseases and injuries. Stem cells can potentially replace damaged or diseased cells. Regenerative medicine. They may enable organs and tissues to be repaired or grown. Drug testing and development. Stem cells can be used to grow human tissues for testing new drugs. And finally, understanding human development. Studying stem cells helps researchers understand how different cell types develop. Risks for the use of stem cells in medical research and treatments. There's a risk of tumor formation, so if stem cells divide uncontrollably, they can form cancers. There's a risk of immune rejection, where the transplanted stem cells can be rejected by the patient's immune system. Incomplete differentiation, where the cells might not fully specialize into the required tissue. And cost and accessibility. Stem cell treatments are expensive and technically demanding, potentially limiting their availability. Social issues for the use of stem cells in medical research and treatments. Access and fairness. High costs may mean only wealthy patients benefit, raising concerns about inequality in healthcare. Impact on research priorities. Resources may be diverted to stem cell work at the expense of other research areas. And vulnerable patients may be persuaded to pay for untested or perhaps unsafe miracle cures. Ethical issues for the use of stem cells in medical research and treatments. Embryonic stem cells. Using these involves destroying the early stage embryos, which some people believe is morally wrong because they view the embryo as a potential human life. Consent. Ethical concerns about whether embryos donated for research were given with fully informed parental consent. Alternatives. Adult stem cells reduce ethical concerns since they do not involve the use of embryos, but may not always have the same level of flexibility. This is because the adult stem cells can only form a limited number of cell types. However, recent research has indicated that some stem cells collected from the umbilical cord act as embryonic stem cells, and using these would help to avoid some of the ethical issues. Long-term consequences. Editing or altering stem cells raises fears of unintended genetic changes or even the future use in human enhancement rather than medicine. Thank you for watching. See our revision notes on this topic. The link is in the description.